Welcome to the afternoon session. We will be talking about the national, regional core facilities. We have three different speakers. Before we begin, I'm going to go through some of the housekeeping notes and rules. Please silence your phones. Let me make sure I'm done mine. The photography is permitted, subject to speakers' request. If they ask you not to, please don't do it. And then ABR of social media policy is everything can be shared. If the speakers ask not to do, please do not do, and tag it with the hashtag ABR of 2025. Unless noted by the speaker, questions are to be held until the end of the session, but I think the speakers may indicate individually the way in which they prefer this one to go. And I think most of you may have uploaded that meeting app. And all the attendees will receive the evaluation survey. Please fill it. I'm requesting that as a chair and as well as the executive board member, because your views are very important for us to plan the next year's event. And with that, I am going to introduce our sponsor, Ms. Emily Lohman from Claret Bio. She is going to talk about Claret Bio's NGS library preparation kit.
Yep.
Thank you. Emily?
Thank you. Good afternoon. Happy to be here today. So to start off, we'll be covering how Claret Bio's NGS library prep kits can be used to generate reliable NGS data from low-quality samples. So to begin, regional and national cores often work with diverse users and diverse samples. And within genomics cores itself, this can lead to a few challenges to consider, mostly the diverse sample sets and user environments creating inconsistent results, the need for multiple kits for multiple sample types within the same project, and a need for automation and scalability for various throughput. Now, current NGS methods sometimes lack the ability to successfully generate libraries from degraded low-input samples while also maintaining sensitivity and simplicity. And so with Claret, we wanted to develop a method that overcame the quality issues with low-input samples. So we developed a method that allows for users to generate data from both single-stranded, nicked, and double-stranded molecules, which in traditional double-stranded preps are lost, and then additionally capture ultra-short molecules such as cell-free DNA and rootless hair. And so with doing that, we're also capturing DNA ends, which is helpful for cell-free fragmentomics and liquid biopsy samples. So the method we developed is called Seriously. It's a single reaction, single-stranded library preparation method. And it's a very simple workflow. You start with your input pool of DNA. You denature and render every single strand single-stranded. And from there, you add on adapters, splint-ligated adapters directly to the ends without any end repair. And then you amplify via index PCR. So there's single tube, no end repair, no A-tailing. And we've created a modular workflow so to allow users to shear their DNA if they have high molecular weight or higher input samples, our shearing module, along with our Seriously kit for DNA and our Really kit for RNA. And we'll cover these throughout the presentation. So the workflow itself is under 3 hours with just 1 hour of hands-on time and fully automatable. And we know cores are adapting new technologies, and so we've made an effort to get Seriously to work well on additional sequencers and liquid handlers that best fit users and project needs. So going over our first year and seriously with FFPE samples. So oftentimes DNA that requires end repair or additional repair steps early on in the process can lead to downstream issues during sequencing, and the lower input also plays a factor in what gets passed through to QC. So the main difference between a double-stranded prep and seriously is the single-stranded ligation and no end repair and no additional repair module. So when we compare the two seriously to a double-stranded prep using 20 nanograms of formalin-comprised DNA, we see that both preps perform well with the mildly severe formalin-comprised DNA. However, with the severely comprised, we're able to generate higher molecular complexity libraries with 5 less PCR cycles and about 3% less duplication rate. And that's a result of the ability to capture both nicked, single-stranded, and double-stranded templates. And when we look at this applied to FFP prostate cancer samples, we see that with only 500,000 reads, we're still able to generate high complexity libraries. And we don't have time to go into it today, but our method can be used with third-party downstream enrichment and has been used for FFPE, buccal swab, and brain cancer patient tissue. So going into our really prep for RNA, oftentimes degraded RNA won't make it through QC due to degradation and contamination from DNA within the sample prep. And so we wanted to allow users to work with this degraded RNA by undergoing first-strand cDNA synthesis, then adapter ligation, then depletion, then index PCR. So you never generate the second strand, you maintain directionality, and you spend less time working with libel RNA. So going into our really or none with our RNA depletion set. So these are two samples that we looked at that both have short insert sizes when a RIN less than 3. And what we saw when we compared it to a double-stranded prep, that we had a higher duplication rate, or a lower duplication rate than the traditional double-stranded prep, even for the poor quality samples. And when we look further into the genomic composition and mapping metrics, we see that with 5 less PCR cycles, we're still generating more usable bases. And when we look at these usable bases, we see that we have less bias in the mapping metrics for the total uniquely mapped to human versus the double-stranded prep for degraded samples tends to favor the ERCC spike in control. So not all folks work just with FFPE, so we created a globin depletion set for folks working with whole blood. And we took 10 nanograms and 100 nanograms of whole blood extracted total RNA and compared the non-depleted to depleted libraries and saw that we deplete both high and low abundance hemoglobin genes deeply sequenced to 20 million reads. Now, this is just a brief overview of what we covered today, the 4-year, seriously, and really. And this is kind of the takeaway from today's talk, that seriously, because you're able to capture the single-stranded, nicked, double-stranded, and high molecular weight DNA, you're able to generate higher molecular complexity libraries. We've allowed for seriously to be run on automation and sequencing platforms other than the traditional Illumina to ensure that you can work with the tools that you need. And our really for RNA maximizes transcript recovery from low input whole blood and degraded FFPE tissue. And our module design allows you to use the resources and tools within a kit that just best fit the samples you're working with. So that's kind of the time we have to cover this, but if you look at the Venn diagram, we do have various applications and sample types we work with other than what was just covered here today. So please stop by our booth and we would be happy to talk more. And we also have a trivia game, so it's pretty fun. Yeah, thank you.
Let us thank Emily for sharing what the Claret Bio can do. As she said, please stop by her booth. And then for today's, my name is Somasundaram. I run a single solo core X-ray crystallographic structural biology core in Florida State University. And of course, I work with Alexei, who runs the National Synchrotron facility. And my colleague here, David, is in Tallahassee, part of the National High Field Magnetic Field Laboratory, or it's called MagLab now. And then also Jeff here runs the NYSPC electron microscopy facility, which is, even though I am a core facility manager at a university, whenever we think of cores, we think of it as either inside an institution or within a small university or a nonprofit institution. We've been trying to expand to within the state, but there are certain restrictions when we go beyond the state. However, what we fail to recognize, there are some regional, national facilities. They are really, really large compared to us. But sometimes we do not think of them as core facility. For example, David has international customers and probably 1,000 people or more they use his facility. And therefore, I thought it'll be nice for us to invite them and share, because the problems they face are the same problems we face, right? All the issues of, and especially in this current situation, we may want to hear from them how they tackle what the opportunity they provide for us. For example, my X-ray facility is, half of it is shut down because I'm unable to compete with Alexei's beamline, which is synchrotron 100,000, 10,000 times more flux than I have. However, he helps me a little bit with allowing me to expand my services. Therefore, I thought today we'll hear from each one of them, their take on the opportunity they provide as a regional and national and even international core facilities, and the challenges how they face and how they are solving. And this is also maybe a good time for us to learn about this and find expanding the facilities, services we provide to other state level or interstate level, and that's why I invited them. They are going to each talk, and then they will share how they want to take the questions or not. And with that, I'm going to, I think David is our first speaker. I'm going to invite David to give his talk. Thank you, everybody.
Okay, thank you very much for the introduction, Soma. Here we go. Hi, everyone. I'm David Butcher. I work at the National High Magnetic Field Laboratory. This is my first time at ABRF, and it's been a great experience so far. I've really learned a lot. There's really a lot about core facilities that is unique compared to my facility, which is actually an NSF major research facility. For those who are not familiar with the MagLab, the National High Magnetic Field Laboratory, or just MagLab for short, like I said, is an NSF major research facility. We have the goal of providing access to the highest magnetic fields for a global user community from a variety of different research disciplines. Well, we're spread across 3 different sites, which you can see here. We have a facility at Los Alamos National Laboratory in New Mexico. We have 2 facilities at the University of Florida in Gainesville, and our primary location, which is pictured here on the right, which is at Florida State University in Tallahassee. We're not really a traditional core facility, not that there is such a thing, but instead we're more of a collection of 7 distinct user facilities, all of which focus on different instrumental techniques and fields of research, but we share the common element of high magnetic fields. Oh, right, I forgot about that. The facility that I work in is the ion cyclotron resonance user facility, which is highlighted here in red, where we do ultra-high resolution mass spectrometry using Fourier transform ion cyclotron resonance mass spectrometers, but we also have facilities that focus on a variety of other different techniques. We have the DC field facility, which specializes in continuous fields up to 45 Tesla. We We have facilities that perform electron and nuclear magnetic resonance. We have an advanced magnetic resonance imaging facility. This is at the University of Florida, where they do a lot of MRI imaging. We have the high B/T facility, also at UF, which does very, very long experiments at very, very low temperatures and high magnetic fields. And we also have the pulsed field facility at Los Alamos, which specializes in short, ultra-powerful magnetic fields up to about 100 Tesla. And we have many users which come to our facilities to take advantage of our unique instrumentation. In 2023, we hosted over 1,800 users, representing about 340 different universities, government labs, and private companies around the world, which gives us a very large impact on high magnetic field research. Here on the left, you can see a breakdown of our users' research disciplines by facility. Some facilities, such as the DC field and the pulse field facilities, which are on the left of that figure, have a high concentration of material science, condensed matter physics research, engineering, things of that nature, while the magnetic resonance facilities and ICR attract more chemistry and biochemistry users, as well as biology, biophysics, some engineering. So we have a very, very diverse group of users overall. And as you can see on the right, our facility is primarily funded by the National Science Foundation through a cooperative agreement with the NSF's divisions of materials research and chemistry. We also receive additional support from the state of Florida and from research awards to individual investigators. Our Amherst facility at UF does tend to serve a larger number of NIH-funded users since they are associated with the McKnight Brain Institute. And the Pulsed Field facility is hosted at Los Alamos, as I said, which is DOE funded. So we have facility support and user support coming from NSF, NIH, and DOE to varying degrees. So to give you an idea of what our magnet systems look like, I'll just quickly show off two of them, one from DC field and one from ICR. In the DC field facility on the left, this is our 45 Tesla hybrid magnet composed of a superconducting outsert magnet and a resistive insert magnet. This was formerly the world record holder for highest continuous magnetic field, but is now tied with a system at the Chinese High Magnetic Field Laboratory. In my facility, ICR, we have on the right a 21 Tesla Fourier transform ion cyclotron resonance mass spectrometer that is also a world record holder and the highest performing system in the world for ICR mass spectrometry, with leading performance in applications from proteomics to complex mixtures analysis. And all of our instruments are available for qualified users from around the world with a competitive proposal process. So in addition to being a faculty member in the ICR facility, where I do primarily top-down proteomics research, my role at the Mag Lab also focuses on data management and facilitating fair data and public access for all the data collected in our facilities. So fair data has become a popular topic over the past few years. It's about making data findable, accessible, reusable, and interoperable for both humans and machines, and also about facilitating reproducibility, which was covered in a couple of different sessions yesterday. Public access became a hot topic after the release of the White House Office of Science and Technology Policy Nelson memo from 2022, which many people probably heard about, and then subsequent updates to public access policies from NSF, NIH, DOE, and all the other federal agencies, which went into effect relatively recently at the beginning of this year. Because we are an NSF major facility, we are obligated to enable users to share their data, to make it fair, and to make it publicly accessible. And this goes along with other obligations, such as ensuring the integrity of the data and being able to track its provenance, which come with the territory of being a user facility. So in order to fulfill these obligations, we have a few different things that we'd like to be able to do. We'd like to ensure that all data collected by our users is retained by the facility in which it is generated, which is an obligation many core facilities have. We want to ensure that that data is indexed, cataloged, and properly annotated with rich metadata to facilitate its reproducibility. And we want to ensure that all that data and metadata is made publicly available at the time of publication, which is the mandate of the Nelson memo and public access plan updates. However, we do have some significant challenges when it comes to actually fulfilling these obligations. First and foremost, we have fragmented and inconsistent data management practices among our 7 facilities. In general, data handling is and always has been very laissez-faire at the Mag Lab since it opened in the early 90s. It's typical in many of our facilities for PIs to use the instrumentation themselves, sometimes with purpose-built instrumental setups, which are only in use for that particular experiment and which cannot necessarily be recreated in the future. PIs can either collect the data themselves, sometimes using their own systems, or be provided data by facility staff and then oftentimes perform their own analysis, which is different than how many cores operate. In these cases, the Mag Lab may not retain a copy of the data at all, or at least we did not in the past. Or we may have the data without any of the metadata that will be needed to contextualize it and understand it, which does not facilitate reproducibility or fairness. We also have a huge problem in terms of the lack of cyber infrastructure, computer systems, data organization systems, and we also have a lack of the necessary human and financial resources to actually build or implement the kind of cyber infrastructure we need. We generate a lot of diverse data across our 7 facilities, which come from both custom-built and vendor-provided hardware and software in a variety of different open and closed data formats. This is a problem that a lot of cores have, but it's multiplied because we're made up of many different constituent facilities. Currently, the Mag Lab doesn't have a system for cataloging or indexing our data or metadata, so everything tends to be stored in kind of a haphazard fashion in large network drives. We do have systems that keep track of the magnet time that's been assigned to a particular user, and that can be used to report the publications derived from those experiments, but the data and metadata are often up to the PI to manage, and we don't necessarily have a record of any of it. We'd like to be able to solve this problem, but the diversity of our data generation means that it's difficult to have a one-size-fits-all solution for data management that doesn't require a lot of manual curation by users, which is not something that they're generally willing to do and will, in fact, actively resist. Concurrently, we're also generating a greater volume of data every year, which is a very familiar problem for many core facilities. My facility, ICR, is especially guilty of this because the mass spec data that we generate is often orders of magnitude greater than many of the other facilities at the Mag Lab. Recently, just last month, actually, some of my colleagues did a series of experiments for a user where they generated a few terabytes of data a day for over a week. There are some major facilities, and I think maybe even Soma's facility, for which this doesn't sound like a massive amount of data, but we're collecting data using desktop PCs that were never intended to have these capabilities. They don't have very large hard drives, and so we were forced to kind of offload the data every night off of the system after business hours to avoid congesting the network. So the data, our ability to generate high-quality data is increasing, which is both a good and a bad thing, but it's outstripping our ability to store it, to process it, and there's no immediate solution that we have ready to implement. Finally, we have a challenge in implementing cultural changes in the established fields of research. The various disciplines of research at the lab that I mentioned before, everything from condensed matter physics to biochemistry, have radically different attitudes and practices around data sharing. Many of the cores here are biological in nature, and anything biologically related, people are far ahead of some other fields in terms of willingness to share and having the tools to share effectively, or to manage data at all. Many of our researchers, and not to call anyone out, but it's particularly the physicists and material scientists, they're far less willing to share and are not really expected to because it's not a norm in their field of research, or for even the publishers of their journals. So this is something that can also be really hard for us to police, both because we have a very large number of users and it's not really practical for us to tell them to do something that they don't have effective tools to do in the first place. We can ask all we want, but if they don't have the tools, then they're not going to do it. Fortunately, public access policies from funders, like the public access plans that have started to go into effect this year, are starting to change people's expectations about what's required of them, which is having positive effects for us. So that's a lot of challenges, and I can't really address all the possible solutions in the time I have remaining. but I'm encouraged by the fact that in terms of cyber infrastructure, there are already a lot of great solutions to handling data management problems, some of which have been discussed here in various sessions, and that are being implemented in core facilities and user facilities of all kinds. I've had conversations with other NSF major facilities, and several of them were in similar situations to the Mag Lab relatively, maybe 5-10 years ago, not that long ago, but they were able to develop a plan and implement the cyber infrastructure they needed and greatly improve the situation. Although many of them are generating a much more limited number of data types, which makes the problem a lot more tractable. We also don't, like I mentioned before, don't have a lot of extensive resources to build cyber infrastructure for data management. So we're trying to form partnerships with other facilities, such as other NSF facilities, to try to implement some cyber infrastructure to address these challenges. We're going to be starting in my facility in ICR. We're working on collaborations to help us build a database, concept of which is shown here, which can be used to aggregate all of our data and metadata, to provide access for project collaborators, which is key. We want to have wonderful things like cloud-based analysis and visualization. We want to have high-speed access for high-performance computing so we can implement big data techniques. After trialing in the ICR facility, we hope that we can roll out some of these systems or some of these concepts to other Mag Lab facilities as well, so that we can have a positive impact on those research communities as well. And I'm obligated to tell everyone at every meeting I go to that we're open to all qualified researchers, so if you want to know more about submitting a proposal, feel free to visit us at nationalmaglab.org, and this is also my contact information if anyone wants to ask me any questions directly. Thank you very much. And if anyone has any questions that they'd like to ask now, I'm happy to take them because I got a few minutes remaining. Okay, we'll save it for the panel then. Oh, do we have one in the back?
There's a microphone.
Oh. It's okay.
Great presentation. Thank you for sharing. We're certainly all. Pardon? Can you hear me better? Thank you very much. Great presentation. Appreciate it. We're certainly all exploring some of these logistics within our various facilities. I was wondering if you were actually planning on making sure that all of that data is immediately publicly available and really thinking about who owns the data at what time point. And I encourage you to be very thoughtful and intentional about this. From my perspective, I never want to be responsible for the data that somebody independently acquired on my facility. My responsibility is to ensure that that instrumentation is working at peak performance. That's my responsibility. If it's a service that I'm offering that they've paid for, I'm now part owner of that data, and I have a responsibility to maintaining it. But I think we need to be very intentional and thoughtful about what we're doing as a shared resource, because that long-term responsibility for that data and the privileged information that's contained therein, I never want to be responsible about having to display that to the public when that might be the beginning of a patent, the next publication, the next grant that goes out for a researcher. They own that. I do not, and I don't want to own it. So I guess food for thought and comment. I think we need to be really thoughtful and intentional about those logistics.
Yes. We've had a lot of discussions internally about data management policy. We have very clearly spelled out terms in our data management plans. We have a data management and sharing plan, which is required by NSF for our whole lab, but we've also written one for each of our individual user facilities. And our policy, I mean, like you said, we don't want to have responsibility, and we're very clear that the responsibility and the ownership lies with the user. Right now, we provide for an embargo period, saying that a user is obligated to make their data publicly available at the time of publication or after 3 years. Now, 3 years is a relatively arbitrary amount of time, but we decided that that was more than long enough for the vast majority of users to have a reasonable opportunity to publish their results.
I think there's an additional stipulation for federally funded research that within 6 months of the end of that funding period, that data must also become public knowledge, which represents some additional logistical headaches. How long does it take to get that manuscript out or the next grant published before you give out that privileged information? So there's some other nuanced elements there as well for navigating that space, I believe.
Yeah, and there's also nuance in just how the documents like the NSF's PA-PPG, the Proposal and Award Policies and Procedures Guide, is written. Sorry, I had to memorize that because I didn't want to trip up on it.
Trying to forget, it's painful.
Exactly,
it's a completely different process, yes.
Yes. So our responsibility as the facility where the experiment is being conducted is to facilitate and enable the sharing of the data, which ultimately doesn't give us responsible for its eventual deposition or public accessibility, but we have to facilitate and enable. And there are other documents like the Public Access Plan, which give us slightly different wording, but we're not 100% responsible for doing that. It falls to the PI.
Thank you so much. Appreciate it.
Hi, my name's Maria Bewley. I'm at Penn State Hershey. I'm also a member of the PRT for structural biology at Brookhaven National Lab. While you may think that 3 years is ample time for data to be released, if you think back to the structure of the ribosome and how many years it took for those structures to be determined, 3 years is insignificant. How do you protect the rights of the graduate students who may be lucky in that they get a crystal structure early on in their career, or they get significant data early on in their career, but require a huge amount of research after that fact if you're only going to give them 3 years to do that?
Well, it's very, very easy to get an extension. What we've also put in our data management policy is if you need longer than 3 years, you simply have to let us know. The other thing is that it's not really being rigorously policed, and we have no intention of going over a PI's head and publishing or releasing data for them, right? If 3 years were to go by, the most that would happen at this point would be that we would probably just politely ask them to fulfill their obligation, or ask them if they intend to publish in the future. So we're definitely not going to do anything without anyone's permission because there would be riots.
Okay, and then thank you for that. How do you get over the fact that NIH says the PI owns their data?
NSF has a similar position. Data ownership in general is a bit of a complex subject for us because, like I touched on, sometimes people come to the lab and use the instrumentation, or they even use their own systems to collect data using our magnet systems. Sometimes our facility staff, and I know because I've done this personally, are integral to the collection of the data, which theoretically, at least given most legal definitions, makes them a data owner as well. But the user doesn't necessarily have an obligation to consider them that way, or to even include them as an author on a publication or anything like that. So there's a lot of gray area here that we're routinely operating in.
So you point to your data, you have a robust data use agreement, and that's your bottom line?
Yes. Now, there are areas where it needs to be improved, but we don't want to be too strict, because then we're going to cause some of the problems that you've already alluded to.
Okay, thank you.
Thank you, Dave.
Thanks, everyone.
I think it's you, I think. This one.
Good afternoon, fellow ABRFers, and thank you so much for the invitation. I guess I'm going to be a harbinger of exactly the opposite perspective that we just saw. A harbinger of realities that affect us as a large facility first, but they're coming for you. I'm here to talk about CBMFs. It's a large facility at Brookhaven National Lab in Long Island, New York. We do crystallography work, structural biology work for international users from all over the world, as well as national users. We're funded through NIH and DOE joint funding at about half-half. We're a fairly large group. There's 6 beamline scientists and 6 permanently associated technical staff, as well as 6 other sort of floating staff and additional management, other support. So it's a little bit more than 20 people. Our challenges, Soma mentioned our challenges. Our recent challenges have been two and anti-parallel to each other. The first is a huge bump in usage demand instantiated by the APS shutdown, the Advanced Photon Source. They're a similar facility to us, and so we had to absorb a lot of their users. I'm not that interested in that one. The second challenge has been a gradual decline in the demand for our facility, which is caused by essentially competition from new technologies. One of them is cryo-electron microscopy, and the other one is AlphaFold and other computer methods. That's what I'm going to talk about, because that's put a lot of pressure on our group and how we responded to that. So the first way we responded to that is by integrating laboratory work that would normally take place in the user's facility into our workflow. So we do that for them. And the way I and some colleagues pioneered this, we used something called acoustic droplet ejection. It's essentially a way to use acoustic methods to harvest liquid materials from a source well and assemble experiments with a lot of flexibilities that come about because of the physical characteristics of sound waves. And the goal here is to move away from the paradigm of structure, which is what we normally do, but which is now we have competition in that. And the paradigm of structure is a user brings us a crystal, which is just an ordered array of protein molecules. We shine some X-rays through it. That produces a diffraction pattern. It's basically a detector with spots on it. We use some computer tools, and that produces a structure. The problem is that now is mostly being done through cryo-electron microscopy and through computer methods. So we needed to reinvent ourselves and create a new identity. All right, so the new identity is going to be, instead of single structures, it's going to be families of related structures, each of which is perturbed by some method. It can be a pH, water activity, and most of all, it's going to be chemical environment. Sort of fragment screening would be the words that you would have heard most typically. And the reason for that is because chemical environment fragment screening is foundational to the development of new drugs, and so it's kind of the biggest fish in the bowl. And the way we handle that is we use sound waves, acoustic technology, to combine small chemical molecules with the crystals that we grow, and then we solve the structure, and now instead of one structure, we have a family of related structures, all of which are in a slightly different chemical environment. We observe the binders, and that can give some clues to chemical biologists to come up with a potential lead or initial drug. And of course, we had fun with that. We created some, for example, some substrates that can handle lots and lots of experiments, so we go faster, faster, faster. So faster, faster, faster. The next thing we did, we also did this and other things. We tried to be better, better, better. So we created different ways of combining our fragments with the crystals. We had co-crystallization. We had, the details don't matter. We're trying to go better, better, better. And then we also wanted to go smaller, smaller, smaller. So we worked with some companies to produce labware that allowed us to work with really small volumes, saving on protein, helping on users, so now faster, smaller, better. And then we reached even further into the user's space, their laboratory space. Now we just asked them for protein, not even for crystals, and we started doing the crystallization ourselves. So a really cool system called combinatorial crystallography, and if this was a scientific talk, I would tell you about that, but it's not. So I'm just going to tell you that it works, and it's really nice. And we do some nice acoustic magic, and we produce crystals, and we collect data on those crystals, and we got some structures. I'm sorry, this thing is, it's an image, but it's PBP5, which is that's our first collaboration, just to show that this can work. It's kind of snazzy, and it's related to antimicrobial drug resistance, and we worked on another protein, which is, it's a collaboration with a Stony Brook professor having to do with inflammation, and had some nice structures from that. And then we also worked on COVID-related proteins, which created was really a nice challenge because the samples were really, really tiny. And we worked on a whole series of different proteins having to do with bioenergy. Really cool. What's the point of all of that? Just to tell you that now we're on a treadmill. So we're going faster, 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 trying to stay ahead. All right, what next? In addition to going faster, and we also wanted to do experiments that were probed a little bit. We're able to work with lower quality samples. So in this case, we do rasters. That means we use the X-rays to probe not just the whole sample, but to zero in on just the best part of it. Or if there is no best part, we can use a whole bunch of samples through serial crystallography and assemble the data into one nice data set from 100 bad data sets. So again, we're trying to extend our facility to be able to take in a greater range of samples because we're trying to stay ahead of this competition. And we got faster robots, and we got that they can work at in the really faster limits, and we got better grippers so that we can operate our robots at faster speeds. We're really on a treadmill here. In addition to, and the goal here is to remove all of the stop signs on this highway between the instantiation of a project and the final result, and just really go into fourth gear. I can't see how this is going to go wrong. So in addition to going faster, we also just jumped into the driver's seat. So how did that happen? Well, our users became less and less familiar with our software. And so we started creating automation tools. Automation tools that allow our users essentially to do a fire and forget, where they would pick this auto raster or auto loop, and then they would just queue up a bunch of data collection requests and walk away, and the instrumentation, the automated hardware, and the automated software would take care of everything for them. And that's great. That's great because it allows us to have better productivity. I don't have a pointer here, but you can see that the dark green bars are higher than the light green bars because, especially at night, because now that the users have a fire and forget, they can just launch their data collection requests and it'll operate at night. That's great. Yes, or from home. In fact, if we're going to be doing automated, why have the users involved at all, right? So then we started with the standby and hybrid, where the users would just send us their samples. So now we've totally depersonified our users. The only thing we see is a box and their spreadsheet with what their experimental goals are. So now we're going faster, 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 and we're further, further, further from our users. Okay, interesting. What's next? Well, we also wanted to help our users with their data processing. And initially we did this with the usual things. We reduced their data for them. We solved their structures for them. My part of it was clustering, so I told you we're producing families of related structures. Well, you don't want a family photo, which is just the average of all the family members. We want to be able to partition each of the different data sets so that we get, family member A, data clean for that, so we get a clean picture of that family, and then B, and then C. We don't want an average of all the participants in a dinner party. We want each of the different people separately, right? So that's what the clustering is about. But anyways, and it works very nicely, blah, blah, blah. We give our users a nice histogram so they can pick the level of granularity that they want to cluster the data at. All very nice tools. The point is we're reaching further and further into their data. Now we've got annotated data, not just anonymized data. We've got annotated data because we prepared their samples for them, and we're processing their data for them. Not only that, now this one, in addition, we've got now families of related structures. So we could interpolate. We could extrapolate. That means we can begin to predict the results of experiments they didn't even do. So that's what this was. We use polynomials to essentially model their data, and then we can use those same polynomials to produce data sets that were between measured data or even beyond the edges. So that brings me to why I'm actually here. Faster, faster, faster. Further, further, further from our users. Closer, closer, closer to their data. You can sort of see the theme here. And so that brings me to my first question for you, which is, have any of you ever watched the HBO miniseries Chernobyl? All right, some people have. Well, in the Chernobyl adaptation, the Legasov, the person who finally blows the whistle on what happened and what was the root cause of the kaboom, mentions, he uses the phrase in the theatrical version, he uses the phrase, every lie we tell incurs a debt to the truth. Now, the real life Legasov didn't say that. He said, we told the truth, just not the whole truth. By the way, what did he mean? He meant that the dangers inherent in the RBMK reactor design, that's the kind of the kaboom, were known locally. Each of the different operators of these reactors had figured out that they had limitations, they had problems, they had safety issues, and they learned those lessons, but locally, not globally. Why is that? Well, because the Soviet Union at the time had the habit of whenever you made a mistake, they'd send you to Siberia, so suddenly there were no mistakes. Nobody makes any mistakes in the Soviet Union. That means nobody was learning any lessons. So that brings us, every lie we tell incurs a debt to the truth. And that brings us to us. What does this have to do with us? Well, we have in our hands, we are facility managers. We have in our hands digital data that do not belong to us, and we have human hands that make mistakes. Those are the ingredients of our own little potential kaboom. So human hands and data that we curate. The problem is that science is not data. That's the whole problem I have with these embargoes and ownership. I think it's wrong-headed to even ask who owns data. It doesn't matter, because data by itself has no value. Science equals data plus sample preparation metadata plus subject matter expertise. If you try to cut the user out of the equation, you end up with science equals data, and that's a catastrophe waiting to happen. You can take it from me. Our greatest asset is not the data piles that we sit on, it's the trust of our users. And when I talk about data science, by the way, just to clarify, because in this community, I know most people think of AI tools and the misuse of AI tools as having to do with a single data set. This was so foreign to me, I didn't even think to clarify when I wrote my little pamphlet that I handed out to a lot of you, that I was talking about something different. I'm talking about training AI on, I'm talking about data mining, when you train an AI on the vast amounts of data that we curate on behalf of our users. Totally different problem. They're both serious problems, but it's a totally different problem. So when you're doing data mining, either through AI or through any other kind of data science technique, the issue is, are you keeping the user in the loop? Because science requires metadata, and it requires subject matter expertise. Otherwise, it's just self-delusion. So how can you protect yourself and the trust of your user? Well, it's kind of embarrassingly simple, really. You already have procedures, I'm sure, for managing risk in cutting grass and buying pencils. You use the same thing. Anytime somebody's going to be performing a data mining experiment on the data that you curate on behalf of your users, but that does not belong to you, you curate it on behalf of your users, you want to have a protocol. You want to have a data policy statement of some kind, and you want to have the 4 tools for enforcement of that statement. The first is you want to question. You need to identify projects. You need to identify when a new project starts. You need training so that people know what to do. You need documentation to back up the training, and you need a safety officer or someone, a person you can call if you have a question. That's how you protect yourself in your individual RBMK reactor. How do we protect ourselves as a community of different operators of reactors so none of us go kaboom? Well, basically that means getting the documentation and the training to be correct. So I got this information from an unexpected source, which is from the early days of aviation. New technology, lots of promise, lots of peril. Initially, the planes were all crashing into each other, into mountains. We had to learn how to fix the problem. How do they do it? It's a 4-part solution. The first is vigilance. We, as a community, have to be aware of potential problems that happen when data mining efforts, well-intentioned data mining efforts, result in something unintended involving the data sets that we curate on behalf of our users. The second one is hard for me, forgiveness. This was what went wrong with the Soviet Union. Nobody volunteers a mistake when you're holding a hangman's noose, okay? So we have to forgive each other when something goes wrong and someone volunteers. The next is flexibility. You can't fall in love with your rules because the rules are only as good, the rules would be the training you make, the documentation you write, and whatever the policies are of the safety manager. Those rules, those practices, those policies are only as good until the next incident. We have to be willing to constantly adapt. And finally, the hardest of all, we need transparency. Each of us in our little RBMK reactors have to be willing to say, something went wrong and we learned from it, and not be ashamed of saying that. Because this is a new technology, and the way you learn about new technologies and their risks is collectively. We can't each make the same mistakes in isolation. We have to learn from each other collectively. And so as I say goodbye, I'm going to make one final plea to you. Please, let's not do this the way the Chernobyl operators did. We all will make mistakes. Let's admit to them, talk about them, learn from them together. Thank you. I can take me back. Huh? Oh, if anyone has any questions, please go ahead and ask. Okay. Thank you.
Thank you.
Oh,
Oh, there we go. All right, well, thank you for the invitation and the chance to talk today. My name is Jeffrey Kieft, and I am the Executive Director of the New York Structural Biology Center. And what I wanted to talk about a little about today is to give a little bit of a, 50,000-foot view of what I've seen over the last 1.5 to 2 years since I've been director of NYSBC, and how it has contrasted with my experience running other cores before I got to NYSBC. So for many years, I was a member of the faculty at the University of Colorado School of Medicine. I ran a research lab there, and was in charge, was kind of the Uber director of the structural biology cores there, the X-ray crystallography core, NMR core, cryo-EM core. We had a proteomics and a metabolomics core. And those were cores that largely served the campus on which they were located. And then when I moved to New York, I took over this other facility, which is clearly a regional and a national facility. And in that transition, I've been able to observe a few things that I think, could offer some lessons learned or some things that, regardless of the size of the core that you're running or the number of users that you have or whether you're regional or local or whatever, we can all adopt. And you've heard some of them already today, but I'll build on those a little bit, and hopefully there'll be some nuggets in here that will resonate with you. So NYSBC is located in New York, as the map indicates, and as the name might clue you in on. It's located in Manhattan, and it was formed a little over 20 years ago by these 9 institutions in and around the New York City area. And that itself was an interesting, I think, genesis, because if you look at the names here, these are 9 institutions that, I don't know if an institution can have an ego, but think fairly highly of themselves. They like to compete often with one another, and yet they were able to get together and collaborate and cooperate and create this center. And so I direct that center, and we're largely independent. So we are on neutral territory. We are not on any of these campuses. We have our own building. We receive funding from all of these institutions, and we provide service to all of them as well. And so that in and of itself now introduces an interesting question that you've already heard about, or an interesting problem, in that we're already facing diversity of users and a lot of different expectations for the people that come from all of these member institutions. Okay. So over the last 20 years, and this is well before I got to NYSBC, it has expanded from a facility that was initially dedicated to nuclear magnetic resonance to now include X-ray crystallography, cryo-electron microscopy. We are expanding into more mass spectrometry and tomography, so trying to work on techniques and offer service in situ structural biology. And we serve a diverse, like I said, a diverse set of users in terms of both the techniques they want to access, as well as where they're coming from and their level of expertise. And I'll point out that we're now at the size to where, as you can see up here, we have 9 NMR spectrometers, so they range in field strength from 500 to 900 megahertz. We operate a beamline right adjacent to where Alexei's beamline is at the National Synchrotron Light Source at Brookhaven. And we have now an electron microscopy facility which has, I think, more than 15 electron microscopes. The director of that department claims we are the largest collection of electron microscopes in the world. I don't know if I'm quite sure that's true, but we're one of them, right? And so it's really grown to be a very, I would say, a place with a great amount of diversity in terms of the techniques that are offered. And I'll also mention that in addition to the 9 member institutions that provide us funding, we also run several national facilities that are funded by the NIH, which are tasked with providing service to people nationwide. And so you can right now see I don't want to say I have a lot of bosses, but I have a lot of people that expect things from me, expect things from the facility, and we have to meet those. And so this gives you an idea, like David was saying, we have people coming from all over. So these are just the users that come for the cryo-EM portion or department within NYSBC. And you can see we have hundreds of projects that we are supporting from users all over the country. So what does this mean? When you go from a facility that is primarily supporting an individual campus or maybe a couple universities, and you have to now start thinking regionally or nationally, what changes? And some of those are obvious, and some of them are less obvious, because you might think at first glance, well, if we're going to now start serving a larger region, we're just going to get bigger, right? It's going to be bigger, but it's going to be more of the same. And I think sometimes, if you run a core facility, you may even be getting pressure from leadership on your campus to do more, right? Bring in more users from a wider area. It'll be fine. It'll mean more in revenue. It'll mean more impact. We're just going to get a little bigger. And in fact, things change in a more profound way when you start to get bigger, for lack of a better way of putting it. And one of the primary things that I have noticed when I compare where I was before and where I am now is that, at a local facility, you obviously have a smaller user base, and this means you can give more personal attention to the users, right? Those who are running the facility or working in the facility can have the time to be highly collaborative and flexible with their time and flexible in how they meet the needs of each individual users. And there's less logistics involved, right? The person who is using your core facility may just be 2 floors up or a building over. And so it's easy for samples to arrive, it's easy to talk about the project, it's easy to schedule time. Obviously, when you move to something that is more regional or national, now you've got a much larger user base. And as you've already heard from the previous 2 speakers, this means a lot of different expectations and diversity in what they want from you. It also means you have to start getting higher throughput, right? You have to think about, how do we manage all of these different samples? How do we find enough time to process all of these samples? And the staff, therefore, it's harder for them to be involved in each individual project. I hate to say it has to become a little bit of a factory, but you start to have to think about, how do we take on a model where we are processing lots of samples, meeting these diverse needs, and all of the additional logistics and things that come with it. So this creates a tension in my mind. You have a user base that's even more diverse than you had when you were local, but you have less ability to meet the individual needs of those users. And so it's a tug of war. How do you meet the needs of all these people who have all of these different levels of expertise and different samples and everything, and you don't have enough staff time to do that, right? Not every staff member can be collaborating if you have hundreds of users. It's just not possible. So how do you adapt to that? And how do you still stay effective? Well, these are just a few, not necessarily too profound observations that I've made or things that I've seen. And I think they're applicable to regional facilities, national facilities, but also perhaps to some local facilities as you grow and you start to think about the challenges within your facility as it changes. So first, you have to come up with some ways to standardize access. It can't just be someone walking in the door and say, hey, I have a cool project. Can we work on it in your core? Will you help me out? You have to kind of come up with pipelines for entry into your facility. It might be proposal-based. It might be different types of access. So for example, I'm using here just from the website for the National Center for Cryo-EM Access and Training, NCCAT, that we run at NYSBC. And there are, users can say, well, what I really need is training. Or I already have a great sample, all I really need is time. Or I think I want to try this cryo-EM and I have a pretty good sample, but I'm not sure it's good enough. What I need to do is come in and make grids and screen, right? And so that way we can kind of funnel people into specific access, and then they have an idea what they have to show up with. What should their sample look like? What can they expect from that sample? Are you really ready for this type of access? It means you're a little bit less flexible, and you're forcing the user to do a little legwork beforehand. But again, it's what you have to do, I think, when you start to get to that type of facility. You do want to get your users, however, to a certain level of knowledge often. right? Because if you're asking them to have a sample in a certain state before they get there, you have to think about, are they going to be able to do that? Or by demanding that, are we going to turn away a lot of users? And so training became actually a large part of what NYSBC does. We have specific programs where users, without even coming in to use the facility yet to gather data on their own samples, can come and get training. Or they can get what we call merit badges. So the merit badges are actually a program we set up with the other national labs where you can get a certain level of training and then you get a merit badge that says, I am now qualified to use the VitroBot. And you can go to any national center and use the VitroBot. Or I am now qualified to mount grids, or I am more qualified to put a sample into the microscope. And so we get them to that level of training. And that means now that they're better equipped to use the facility because we kind of put in the legwork or the time beforehand to get them trained up. And these are very, very popular programs that we've come up with. As you start to set up training programs and you start to turn in a little bit of a factory and you start to have more specific gateways for access to the facility, you're going to have staff that deal with those things specifically. And what I mean by that is you may have to have staff that are dedicated specifically to training. That's what they do. They don't handle samples. They're not really operating the microscopes or the NMR or whatever it is. They're just setting up training programs, doing workshops, getting feedback, working with users in that way. Of course, you need the technical staff. You may need the maintenance staff, because if you're trying to have 1 or 2 or 3 people and they're wearing every hat, it really doesn't work. And so even I think at smallish facilities, or if there are multiple core facilities at a certain university that are doing different techniques, having someone that's spread between those 3 or 4 core facilities that sets up training programs, they're kind of the curriculum development person for the cores on campus, can be incredibly valuable. And it also gives you another reason why you are important to the campus community, because you're offering training, right? You're doing more than just running instruments. You're providing that kind of enrichment for the campus community. And one thing I did not list here is IT. I think we've talked a lot about IT and data, but this is a huge problem that we also face, is just capacity. What do we do with all the data? Because one microscope can generate 4 to 5 terabytes of data at a single session, and we've got 9 of those running full time. So you can do the math. We're building up tens of terabytes or more of data a day, and we can't possibly store it all. And so that creates an interesting problem, which I can talk about in the Q&A if you're interested. Whoops, did I go too far? But the last thing I'll say is that I also think it's important, though, as one, I've made it sound very much like a facility like NYSBC becomes a factory. And I don't like that word because it makes it sound like all the staff are doing are, they're like in that I Love Lucy episode. Have you seen that, right? Where Lucy can't keep up and she's just trying to put the chocolates in the little thing. You don't want it to become repetitive or boring for highly skilled, smart people that have a lot of training and are scientists at their core. And so finding ways for the staff to collaborate sometimes on projects that are of interest to them or where there's high value, or allowing them to have research programs or projects that help to keep the facility at the cutting edge, I think is really important. I mean, there have been a number of technologies and processes that have been invented at NYSBC by the staff that are either patented or disseminated or more widely used throughout the structural biology community. And that helps to create, a vibrancy and an excitement among the staff. And also, by having the staff do research on interesting problems, it means that when your users show up, they can ensure that they're getting the highest, most cutting-edge expertise that's available, right? Because they may come in, users from around the region, and say, hey, I saw this technique in a meeting, and this looks really cool. I want to do it. Can you guys help me do it? And you want your staff to be already there saying, yeah, we've tried that, or we're working on that, or we've got an even better way to do it. And that, again, I think is really important. And it helps keep the staff around because, if they get bored, they may decide to go elsewhere. And that's a constant problem, is turnover of highly skilled, really well-trained people. So that's the last thing I'll say. And again, these are kind of just general things that I've noticed. But I guess my take-home message is, when you grow, you're not just growing. You have to think a little bit differently, and you have to think about how do the changes that come with expanding into a regional facility or even just a larger facility on your own campus that is going to try and serve a larger block of people. What are the inherent challenges that come with that are more than just more of the same? And I'll stop there, and I'm happy to take any questions. I got the microscope all the way at the front, the microphones. That takes
Hi, thanks. So it's really interesting. Made me think of a few things. So the first is, can you talk a little bit about, I guess, education separate from training and also consultation? So how do the PIs know whether, like, I've got something small and floppy, how do I know if it'll work with NMR or if I want to, freeze it? Or maybe it'll crystallize if we're lucky, or if we substitute, like, how do you work together with people at that level?
Yeah, that's a good question. So, it's interesting when I, and this, hopefully I can answer this. When I first got to NYSBC, it was interesting, the X-ray crystallography, NMR, and cryo-EM departments were operating largely independently. And there was kind of no go-to person that you would go to and say, hey, I don't know much about structural biology, but I have a structural problem. How should I even approach it? No one to really consult with. You kind of had to know what you wanted to do, and then you went to that department. That's changed a little bit. We started to expand what I call the special projects group, and that's a group of highly skilled scientists. They don't belong in any one of those departments, and they can do that kind of collaborative work or consultation. You just kind of approach them and say, here's what I want to do, how should I do it, and you kind of develop a plan. So that's, maybe that kind of answers your question. And sometimes it's me, quite honestly. I'll run into someone at a meeting and I'll say, drop me an email and we'll figure it out. Education and training, yeah, I kind of mix those together. And in terms of like the traditional didactic lectures, graduate student education, we actually do some of that. We actually run classes at NYSBC that the centers use as part of their graduate school curriculum. But the training, I think, is most valuable for the users that come in when it's really a hands-on kind of training, right? They come in, they learn some of the theory of, let's say, cryo-EM, if that's what they're going to do, and then they get their hands on the instruments, and we get them to a certain level of competency. And I should say there are actually, one can write a proposal and get access through NCCAT, and a PI can come with, say, 2 of their graduate students out for 2 weeks and get an intense hands-on training experience. So there is flexibility. I don't know if that answers your question.
But the merit badges are primarily about technical skill based.
Yes, that's right.
Not
That's right.
Theory, not like knowledge transfer type stuff.
Correct, yes.
Okay.
Actually, Josh kind of hit on my question about your merit badge program. I looked on your website, doesn't say anything about it. Is that something that you would be willing to share, how you structured that? And
absolutely.
How that...
Yeah, so the merit badge program is part of the NIH-funded National Center. And I take no credit for this whatsoever. This happened before I got there. And so in partnership with the other national centers, they got together and said, we should standardize this. And so again, it's partially online, it's partially hands-on. But yes, I can send you and put you with the person that came up with that.
Yeah, that would be great. I think it could be a great resource and a great model for a lot of places to use. We do have in many of our core facilities that type of, the balance between education and training, right? How do you find the time to actually spend, how much time do you decide to allocate towards an education program, which gives people basic information and tools and applications, but then also provides enough time to do the technical training, to be competent with the instrument, know what the instrument does on the inside and isn't just a black box, so that you can interpret the data that comes out of it. And I think that's a both very important program. So, yeah, thanks.
Should I do the panel?
Can I ask another question?
Oh, yeah, sure.
And I don't know, maybe this is a question for both you and
Well-
you. Well, I don't know. Let me just throw it
Yeah,
out
we
there.
could sit down and do the panel.
I'm sort of, I guess I know enough about structural biology, and I'm old enough that I see, like, there have been several revolutions, and I think it's really interesting to me because, like, there was a time, right, where, like, a PhD project, like a thesis project could be, we want to crystallize this thing, we want to get diffraction, and we want to solve a structure, and it's going to take you 5 or 6 years to do that with some help and some expertise and some training. And now we're saying, like, I don't know, I'll send you a sequence and you'll give me 100 structures of variants of it, you know, tomorrow. So, like, where does the PhD student fit? I mean, it's kind of like a woolly question, but where does I'm a big believer in, like, we're in academia, right? Like, most of us are in academia in one way or another. And I'm a big believer in, like, the PhD and the PhD project, and, like, it has meaning to me. So, like, where does the PhD student fit into this, like, high throughput, you know, hands-off, fantastically facilitated from the point of view of going from question to answer? Where do they fit into that? Is that a weird question? I don't know.
That's not
I think it works.
It's not a button. Oh, okay. Well, that's a good question. So I can answer from my own personal experience. So I still am training graduate students, and I have a couple that moved with me to New York. And there's one who is both of them actually are their studies, their dissertations will be based primarily on trying to solve a structure. You know, a lot of it is not just the structure itself, but everything that surrounds it, the interpretation and the sample preparation and trying to figure out what is the right structure to solve. And then even then, you know, they're both using cryo-electron microscopy. It's a bit of a slog to get the good grids and to get know how to process the data. And, you know, now we can see multiple states of the particles on one grid. And so how do I parse those out and what do they mean? So there is still a lot of intellectual engagement, at least there. But, you know, in some cases, I think we are moving. You know, I've said in 5 years, I think cryo-electron microscopy will be a lot like crystallography in that we won't really need a user. You'll put something into the microscope and a structure will come out because it'll be fully automated. And so then I don't know what the answer to your question is.
To answer your question, Josh, I started 33 years ago where all the students were solving their whole PhD one structure. Therefore, they had to do everything from knowing how to mount a sample, how to collect data, how to grow the crystal, everything. 33 years later, half of it is gone. They send it what we call FedEx data collect. They send hundreds of samples, and in Alexei's beamline, we'll collect data. They don't even know what name of the sample is, which basically it's an Excel spreadsheet. We, as a person working in the university where education is the important thing, we really see that as a very sad day for students not even knowing how they got there. But however, the pressure to produce more data, no student can publish one structure, get a PhD now, right? That is the problem, right? That's unfortunately, we are trying to still keep it home, whatever we can do the training, but you are correct. A lot of students are not completely getting trained on how to do all the steps.
Another analogy would be like knockout mice. Like,
Right.
I'm old enough that, like, I know people who their PhD project was to make a knockout mouse,
Right.
and then in 6 months they would try to figure out a phenotype and to get their thesis, right?
Exactly, right.
And that's a means to an end. Like, the structure is the end here, right? Like, I mean, yes, you can say, now that we know the structure, can we design drugs, or can we, look at mutants or variants or substitutions or whatever, but, like, getting the structure is a viable endpoint to a project. Whereas with a knockout, like, I'm fine with people being able to, like, get a knockout mice from Jackson Labs in 6 weeks or whatever, because that allows them to do their science. So it's different.
Yeah. Thank you. Jeff, I mean, I know we are running out of minutes. I'm just going to ask you one question, which is probably a lot of core facility people may feel. There is a pressure to consolidate and make a centralized core. You mentioned that to incentivize that, you're allowing people to do a special project. Should all the core facility, how do you justify, does the staff are allocated like in Google or 5% their own time? how do you justify in their salary? Because many of the core facilities are centralizing now, many of them are asked to do more. How do the individual core facilities instead of becoming a factory, how can they have some special project? Who pays for them? How do you allocate? Could you comment on that?
Yeah, I mean, I can say how we're able to do it, and it in part is in part